Okay, so you have any doubt in first chapter?
No, ma'am.
See, they'll be sharing the question papers. So along with this, you have to solve that also. And if you have any doubt, you note down the question number, and next day you have to tell me. Okay. In the very next class, I'll explain the doubts, because we won't get time for discussing all these things in class time. Okay. Okay. So they'll be sharing the first chapter questions. You try with that. Okay.
Ma'am, I got the question paper, ma'am.
Okay, you solve all those things, and I'll share more questions if I'm getting. And then try with all those questions. With each chapter, you have to solve. Okay. Okay. So we'll move to the next chapter. This is biological classification.
Yes, ma'am.
So in this chapter, we are going to discuss about the classification of organism. So here you can see this screen now.
Yes, ma'am.
Harshini?
Yes, ma'am. Yes, ma'am. I can see, ma'am. I can see the screen, ma'am.
The screen is clear now. Okay, if there is any problem and the voice is breaking, you can tell me. Okay, I'll start now. Here you can see the picture. In this, you can see many living organisms. Here you can see so many living organisms. And how we classified, we know that the objects which are showing growth, then adaptation, reproduction, all these are called as, all these characters, the organism which shows all these characters are called as living organism. So we need to classify all these organisms for our studies. So how we will classify the organism? For that, we are following the biological classification. And what is biological classification? The scientific procedure which we are following to arrange the organism into groups and subgroups. And on what basis we are grouping this organism? Based on their similarities and dissimilarities. So based on similarities and dissimilarities, we are placing the organism into different groups. It is called as biological classification. So we are classifying organism. Which organism? Living organism, which are showing growth, development, responsiveness, then reproduction. All these characters are present in living organism. So we need to classify the living organism. And how we will classify? Based on the similarities and dissimilarities, we are placing them into groups and subgroups. So this procedure, or this is called as biological classification.
Okay, ma'am.
So many scientists, they tried for this classification, but it was not properly done. One second. Next, based on all these scientific reasons and all, Aristotle tried for the first classification of organism. So Aristotle was the first person who tried for the classification. You note down all these points, okay? So it will be easy for your revision. So based on scientific reasons, Aristotle made first classification. And he was the first person who did this classification, and he made classification based on the morphological features. Aristotle's classification was based on only morphological features, and he classified plants and animals. He classified plants into three categories. Which are all the categories? They are trees, shrubs, and herbs. Aristotle classified plants and animals. He classified plants into three categories, which are trees, shrubs, and herbs. And he classified animals into two groups, which are the two groups? With red blood and without red blood.
Without red
Here
blood.
I mentioned only the animal classification, but he classified plants also. He classified plants into trees, shrubs and herbs, and classified animals into with red blood and without red blood. Then after this also so many classifications, so many trials or attempts were made, and next was done by Linnaeus, and this is called as two kingdom classification. You can see here the two kingdom classification, and from the title itself we can make out it is just two kingdoms were present in that, which were there, that kingdom Plantae and kingdom Animalia. He classified, Linnaeus classified in organism, living organism into two kingdoms. First one is kingdom Plantae and next is kingdom Animalia. And he, under kingdom Plantae, he classified all plants under kingdom Plantae and kingdom Animalia, which includes all animals. Thus he made two kingdom classification. Linnaeus made two kingdom classification, and so the important things to remember is two kingdom classification was proposed by Linnaeus and two kingdoms were there. One is kingdom Plantae and another one is kingdom Animalia. And he included all plants under this one, kingdom Plantae, and all animals under kingdom Animalia. Only two categories were there in this. So we can tell as two kingdom classification is inadequate. Only two categories were there. This was having so many drawbacks, this two kingdom classifications. Because here you can see the two kingdom classification in this picture. This represents the two kingdom classification. In this you can see only plants and animals. Just in previous slide we explained only two categories were there, plants and animals. But this was not proper, or this was not correct. It was having so many drawbacks. What are the main drawbacks of this one? This is not differentiating eukaryotes and prokaryotes. Means in under eukaryotic categories, even prokaryotes also, prokaryotic organisms also coming. So it was not able to differentiate the prokaryotic and eukaryotic organism. And same time, it was not able to differentiate the unicellular and multicellular organism. And photosynthetic, not able to differentiate the photosynthetic and non-photosynthetic organism. Got it? It was not able to differentiate eukaryotes and prokaryotes, unicellular and multicellular organism, and photosynthetic and non-photosynthetic organism. So under one group, both types were coming. So this was not a proper classification. But in this classification, what Linnaeus did, he easily classified plants and animals. This classification was very easy to understand. It was easy to understand, but so many organisms did not come under any category, either of this category. Some organisms did not come under either plants or animals. So this was a main fault or main drawback. Because of all these reasons, we can say that this classification is inadequate, or these are the main drawbacks. It was not able to distinguish eukaryotes and prokaryotes, and unicellular and multicellular organism, and photosynthetic and non-photosynthetic organism. And the main problem was so many organisms. We cannot classify so many organisms under either of this category, either into plants or or into animals. So that was the drawback of two kingdom classification. And after two kingdom classification, same like this, three kingdom classification came, four kingdom classification, but all were having the same issues. The classification issues, or it was not based on proper criteria. Next, after all these things, a scientist named R.H. Whittaker in 1969, Whittaker classified, or he proposed the five kingdom classification. In this classification, it was based on body organization, cell structure, then reproduction, then phylogenetic relationship, and mode of nutrition. These are the characters which he mentioned for the, or as the classification criteria. he took these characters as the criteria for the classification of organism. Which are the characters? Mode of nutrition, reproduction, body organization, phylogenetic relationship, and evolutionary relationship of organism. So in 1969, he proposed 5 kingdom classification. MCQ question, note down. Whittaker classified 5 kingdom classification, and 5 kingdoms were there. Which are the 5 kingdoms? Kingdom Monera, Kingdom Protista, then Kingdom Fungi, Kingdom Plantae, and Kingdom Animalia. These are the 5 kingdoms. Whittaker in 1965 he proposed, and based on which characters he proposed? Based on mode of nutrition, reproduction, body organization, phylogenetic relationship, and mode of reproduction. Based on all these things, he classified organisms into 5 kingdoms: Kingdom Monera, Kingdom Protista, Kingdom Fungi, Kingdom Plantae, and Kingdom Animalia. Next, from this you can make out, from each picture you can make out which and all the organism coming under each category. Monera, here you can see single-celled organism. But when it is going to the higher category, you can see advanced levels. In Animalia, and can see advanced characters. So based on the characters, he classified into 5 categories or 5 kingdoms: Monera, Protista, Kingdom Fungi, Kingdom Plantae, and Kingdom Animalia. Here you can see the advanced characters. Next, this is a chart which is showing the characters. Here you can see the characters of each kingdom. And which are all the kingdoms? Kingdom Monera, Protista, Fungi, Plantae, and animals. And here first is the cell type. First character is cell type. In Monera, they are prokaryotic organism. Means there is no nucleus. Cell type is prokaryotic in Monera. There is no nucleus. So next character, nuclear membrane. If nucleus is absent, obviously nuclear membrane is also absent. So you can write this characters easily. Monera, characters of Monera prokaryotic, nuclear membrane is absent. Then what about the body organization? At cellular level. I showed in that picture, in that chart, only cellular level, only single-celled organisms, bacteria and are present in Kingdom Monera. So cellular level of organization. And mode of nutrition is autotrophic or heterotrophic. Autotrophic organism is also present, or heterotrophic organism is also present. They are also present in Kingdom Monera. But autotrophic means they'll be chemosynthetic and photosynthetic. Chemosynthetic and photosynthetic organisms are present. But heterotrophic means they'll be saprophytic or parasitic. Both types are present. Got it? Okay. So in Monera, the cell wall, we didn't tell about the cell wall. Cell wall is non-cellulosic. Means it is not made up of cellulose. Instead of that, it is made up of polysaccharide and amino acid. Polysaccharide and amino acid. Cell wall is non-cellulosic. The character, you should note down this character. It is non-cellulosic. Non-cellulosic cell wall is present in Kingdom Monera. So in Kingdom Monera, they are prokaryotic. Cell wall is non-cellulosic, and this nuclear membrane, prokaryotic cell, it is not having nucleus. So nuclear membrane is also absent. Then body organization is cellular. They are autotrophic and heterotrophic. Autotrophic means chemosynthetic and photosynthetic organisms are present. Heterotrophic means saprophytic and parasitic. Both types are present. Next, we'll go to the characters of Protista. Protista, in this, you can see bit advanced, because here it is prokaryotic. Here you can see advanced level, not slightly advanced. It is eukaryotic. And cell wall is present in some. In some organisms, cell wall is present. But nuclear membrane is present because here it is eukaryotic. Nucleus is present, so nuclear membrane is also present. Body organization is cellular level, same like this cellular level of body organization. Then mode of nutrition, it is autotrophic and heterotrophic. We can see autotrophic and heterotrophic organisms here also. Next is kingdom fungi. You have any doubt in this?
No, ma'am.
Okay, next is kingdom fungi. Fungi means the cell type is eukaryotic. See, from here you can see only eukaryotic cell type. Only in Monera you have prokaryotic cell. Rest and all you will be seeing this eukaryotic cell types only. In fungi, the cell type is eukaryotic. And the cell wall chitin is present. It's with chitin. The cell wall is with chitin and nuclear membrane. Here eukaryotic cell, so nuclear membrane is also present. But here body organization, here in Monera and Protista, you can see cellular level of organization. But when it comes to this fungi, multicellular or loose tissues are present. Multicellular level. From cellular, it is going to multicellular or tissue level, loose tissue level organization. Means the characters are getting advanced in each kingdom. Next is mode of nutrition. This is heterotrophic. Means saprophytic or parasitic. It is not synthesizing. There is no autotrophic organism in kingdom fungi. It is having only heterotrophic organism. And next is kingdom plantae. In kingdom plantae, the cell type is eukaryotic. And the cell wall, everyone knows cell wall in plants, cell wall is with cellulose. Next, nuclear membrane is present. Here eukaryotic, so nucleus will be present, then nuclear membrane is also present. Then here, body organization. From compared to this fungi, you can see little more advanced. Here is tissue level or organ level. Tissue or organ level of organization is present. They are autotrophic, means they are doing photosynthesis. So plants are doing photosynthesis, so they are autotrophic, and tissue level of tissue or organ level of organization is present, and nuclear membrane is present. Then cell wall is with cellulose. Then cell type, eukaryotic cell type. Next is kingdom Animalia. In Animalia, again eukaryotic cell type is eukaryotic. Cell wall is absent. In animals, cell wall is absent. So the cells are flexible. And nuclear membrane. Nuclear membrane is also present. And here in animals, tissues, organs or organ system. Tissue level of organ, or some organisms will show tissue level of organization. Some are showing organ level of organization. Some are showing organ system, like our digestive system. In digestive system, so many organs are present, and they are combining and doing one function like digestion, right? Together they are performing a function like digestion.
Yes, ma'am.
And animals are heterotrophic, means they are not doing photosynthesis. So is there any doubt in this chart? Is it clear?
Yes, ma'am. Clear, ma'am.
Okay. Now we'll go to kingdom Monera. These are bacteria. Kingdom Monera, they are nothing but bacteria, and they are unicellular organism. In the previous chart itself, I showed all these pictures. So from that itself, we can make out they are having only unicellular organism, and they are microorganism. They are most abundant microorganism. All microorganisms are abundant microorganisms are coming under kingdom Monera. Mainly they are bacteria. Here you can see, if you are touching somewhere also, bacteria will be present. So they are present everywhere. Almost everywhere they are present. And the cell wall is made up of peptidoglycan. Their cell wall is made up of peptidoglycan. And the peptidoglycan present in most of the bacteria. Note down this, it's not there in this PowerPoint. Peptidoglycan present in most of the bacteria is N-acetylmuramic acid. N-acetylmuramic acid or NAM, N-A-M. This is important question. And one more is N-acetylglutamic acid. In bacteria, peptidoglycan is present, and the main peptidoglycan which is present in bacteria is N-acetylmuramic acid and N-acetylglutamic acid, NAM and NAD. Next is they can live in extreme no
Your voice is
conditions
breaking, ma'am.
like snow, hot spring. So this bacteria or the organisms of kingdom Monera, and many bacteria they are living as parasites. Many of them are living as parasites. And in the chart, just in previous chart, I have already told they are prokaryotes and they don't have a nuclear membrane. So DNA is naked. DNA is not covered by DNA is not inside the nucleus. So it is a naked DNA, and body organization is cellular level, and cell organelles. Cell organelles, we can all the cell organelles like endoplasmic reticulum, Golgi bodies, then lysosomes, mitochondria. These things are absent. Okay, they have ribosomes. Ribosomes are present in this bacteria. So what are now the important points? They are most abundant microorganism. Is it clear?
Ma'am.
Okay. They are abundant organism and they'll be present almost everywhere. And they are living in extreme habitats, extreme conditions. And they are living as parasites. Then ribosomes are present in them. They have ribosomes. Many cell organelles are absent in this. Okay, ribosomes are present in bacteria. And their DNA is naked. It's not inside a nucleus because they are prokaryotic. Next is, based on the shape, bacteria are grouped under 4 categories. Based on the shape of bacteria, they are classified into 4. First one is coccus. Next is bacillus. And third one is vibrio. And fourth one is spirillum. First, coccus is spherical or round shape. Based on the shape, we have divided bacteria into 4: coccus, bacillus, vibrio, and spirillum. And coccus, it will be spherical or round shape. Then bacillus, they are rod shaped. And the third type is vibrio. Vibrio is, here you can see the coma shape. Bacillus is rod shape. Coccus is round or spherical shape. Next is spirillum. Spirillum is spiral shaped. See, bacteria structure is very simple. Structurally, they are very simple organism, but they are showing diversity in their metabolic activities or metabolic characters, and they are very complex in their behavior. See, bacteria structure-wise it is very simple, but metabolic activities it is showing great diversity. And some are autotrophs because they are synthesizing their own food from inorganic substrate. Some are autotrophic. Autotrophic bacteria are there because they are synthesizing their own food from inorganic substrate. Then majority are heterotrophs. Majority of these bacteria are heterotrophs. They are not synthesizing their food, but they are depending on other organism or dead matter. So structurally, it is simple, but metabolic activities. In that case, it is showing diversity and they are complex in their behavior. And some bacteria are autotrophic, means they're synthesizing their own food, and most of them are heterotrophs. They are not synthesizing their own food. They are depending upon dead organic matters. So next is classification of kingdom Monera. Up to this, it's clear.
Ma'am. Yes, ma'am. Clear, ma'am.
Any yeah. Okay, then let's move to the classification of kingdom Monera. See, they are mainly classified into archaebacteria and eubacteria. Bacteria are classified into archaebacteria and mainly they are classified into archaebacteria and eubacteria. The archaebacteria, they are again divided into halophiles, thermoacidophiles and methanogens. Then eubacteria is classified into autotrophs and heterotrophs. This autotrophs are again divided into photosynthetic autotrophs and chemosynthetic autotrophs. And heterotrophs are divided into parasitic and saprophytic. So bacteria or kingdom Monera, it is divided into archaebacteria and eubacteria. This archaebacteria is divided into halophiles, thermoacidophiles and methanogens. Then eubacteria is divided into autotrophs and heterotrophs. These autotrophs, which are synthesizing their own food, they are divided into two, photosynthetic and chemosynthetic. Then heterotrophs, which are not synthesizing food, which are depending on other organisms, they are divided into parasitic and saprophyte. Clear?
I'm clear, ma'am.
So let's move to the first one, archaebacteria. In kingdom Monera, there are two types of bacteria, archaebacteria and eubacteria. So archaebacteria they are the ancient bacteria or they are the primitive type. They are the ancient bacteria or they are the primitive type. And these type of bacteria, they can live in extreme conditions. They can live in salty areas, then hot spring, then again marshy areas. So they can live in extreme conditions like this. So the bacteria which are living in salty areas are called as halophytes. And hot spring, they are living in hot spring. They are called as thermoacidophiles. Thermoacidophiles. Here you can make out by these terms. Thermoacidophiles means hot spring areas. And marshy areas, methanogens. Methanogens can live in marshy areas. And they are cell wall structures. Their cell wall structure is somewhat different. So that is making them to, or that is helping them to live in such extreme conditions. Their cell wall structure is different, which helps them to live or survive in extreme conditions. Then methanogens. Methanogens are present in the gut of ruminant animals like buffalo, cow, and all that. No? Like in the gut of such type of animals, these methanogens are present. These are animals. These animals will get their food from plant-based food. They'll have plant-based food. And these organisms, which is present in the gut of this buffalo or cow, they will ferment or fermentation will take place in specialized stomach. Before digestion, one kind of fermentation will take place in specialized stomach with the help of this methanogens. And these methanogens are responsible for the methane from the dung of animals. So this cow or buffalo, they will take plant-based food, and before digestion, a specialized in special stomach or specialized stomach, this reaction will take place with the help of this microbial action. This special kind of fermentation will take place with the help of these methanogens. So they are responsible for the methane or production of methane from the dung of these kind of animals. Is it clear? The role of methanogens? Next is the second type. In Monera, kingdom Monera, first is Archaebacteria, next is Eubacteria. Eubacteria are called as true bacteria. These are called as true bacteria. They include autotrophs and heterotrophs. Both autotrophs and heterotrophs are present in Eubacteria. This autotrophs consists of photosynthetic and chemosynthetic. They are synthesized. Autotrophs means they are synthesizing their own food, and they include photosynthetic autotrophs and chemosynthetic autotrophs. And they have very rigid cell wall. Their cell wall is rigid, and flagellum. It is in motile forms. Flagellum is present for locomotion. So in eubacteria, they are called as true bacteria. They include autotrophs and heterotrophs. Autotrophs consist of photosynthetic autotrophs and chemosynthetic autotrophs, and heterotrophs also present in eubacteria. Then, and in eubacteria, the cell wall, rigid cell wall is present, and flagellum is also present in motile forms. Next is the autotrophs are classified into photosynthetic and chemosynthetic. In eubacteria, there are two types of autotrophs, photosynthetic autotrophs and chemosynthetic autotrophs. So first one is photosynthetic autotrophs. Example for this is cyanobacteria. For photosynthetic autotrophs, cyanobacteria are the example. The cyanobacteria is also known as blue-green algae. They are known as blue-green algae. And they are photosynthetic and they have chlorophyll A, which is similar. The same chlorophyll is present in plants, which is helping for photosynthesis. So the same chlorophyll A is present in photosynthetic. Then the cyanobacteria are unicellular and they are present in colonial forms. See, they are unicellular, but they are present in colonial or filamentous form. You can see here. And they are present in freshwater, marine water. In all these, they are present. And sometimes they are forming blooms. In polluted water bodies, it will form blooms. And these colonies, I said just now, I said the cyanobacteria, they are forming colonies. They are found in colonial forms. See, when it is forming colonies, you can see a type of sheet or covering around the colony. This mucilaginous sheet you can see here. It is present in cyanobacteria. So you can see the mucilaginous sheet around the colonies. And they can fix atmospheric. Some of them can fix atmospheric nitrogen. And specialized cells called heterocyst. Here you can see heterocyst, the specialized cells. These are helping for atmospheric nitrogen fixation. So this they are called as blue-green algae, and they have the same chlorophyll A, which is present in the green plants. And the cyanobacteria, they are sometimes they will be forming colonies or they are in filamentous forms, and they are present in fresh water, marine water, etc. But in the colonies, you can see the mucilaginous sheet covered on the colony. And here you can see heterocysts. These are the specialized structure cells which are helping for nitrogen fixation. An example, nostoc, and anabina. These are the examples for cyanobacteria. Note down this examples for cyanobacteria, and then heterocyst is an important question. Then mucilaginous sheet, this is also, and the chlorophyll content which is present in the cyanobacteria. These are the main points. Nostoc and anabina are the examples. This photosynthetic autotroph. Then in eubacteria, next is chemosynthetic autotroph. Now we have learned about photosynthetic autotroph, and they have chlorophyll A for photosynthesis. Here next is chemosynthetic autotrophs. Chemosynthetic autotrophs, they oxidize inorganic substance. They oxidize inorganic substance such as nitrites, nitrates, and ammonia. These chemosynthetic autotrophs, what they are doing, they are oxidizing inorganic substance such as nitrates, nitrites, and ammonia, and they will release energy. And the released energy they are using for ATP production. The released energy is used for ATP production. Their photosynthesis is taking place, and they are utilizing that energy. Here, chemosynthetic autotrophs, they are oxidizing these chemicals or nitrates, nitrites, and ammonia. Then they are using the released energy for the production of ATP. And they have important role. These have the important role in recycling nutrients like nitrogen, phosphorus, iron, and sulfur. Due to sulfur bacteria, you can see this red kind of appearance in some water bodies. Okay, this is chemosynthetic autotrophs. You have to note down in this ATP production from these oxidizing these inorganic substances, and they are producing that energy for ATP production. And next is heterotrophic bacteria. They are abundant in nature. So many are present in nature, and the majority are important decomposers. They are mainly the decomposers. These heterotrophic bacteria are mainly decomposers. Next is the role of heterotrophic bacteria or their impact on human affairs. Here, the role, they have different roles. Some are pathogens causing damage to crops. Plant pathogens are present. In heterotrophic bacteria, plant pathogens are present. This is plant disease like citrus canker. You can see this black dots, that is called as citrus canker on lemon. So that is caused by these, and some are causing damage to human beings like cholera, typhoid, tetanus. All these are caused by these organisms. Then production of antibiotics. Antibiotic production is also very important, and they are making curd from milk, or they are helping to make curd from milk. And some pathogens, animal pathogens are present, and some are helping to fix atmospheric nitrogen in leguminous plants. So what are all the roles? They are, some are animal pathogens, some are plant pathogens, and some are human pathogens are there. Then they are fixing atmospheric nitrogen in the roots of leguminous plants. Then production of antibiotics and helping to form curd from milk. Then next is the reproduction in bacteria. And mainly bacteria will reproduce by binary fission. So when unfavorable condition comes, they produce spores. Here you can see. During unfavorable condition, where the binary fission is not possible or the condition is not favorable, they will produce spores to survive that unfavorable condition. So they reproduce by kind of sexual reproduction. Sexual reproduction by the transfer of the DNA from one bacterium to another. Their sexual reproduction, it's a particular type of sexual reproduction by adopting a primitive type of DNA transfer from one bacterium to another. A special kind of DNA transfer from one bacterium to another. That's the way they are reproducing sexually. But during unfavorable condition, it will form spores to survive or to withstand that unfavorable condition. Then bacteria, mainly they are reproducing by binary fission. So the main method of reproduction is binary fission. And next is mycoplasma. These mycoplasma, they are the organism that completely lack a cell wall. They don't have a cell wall. And they are the smallest living cells. They are the smallest living cells. Now, note down this. Mycoplasma are the smallest living cells. And they can survive without oxygen. This mycoplasma can survive without oxygen. And they are pathogenic in animals and plants. Many of them are pathogenic in animals and plants. So mycoplasma, they don't have a cell wall and the smallest living organism, and they can survive without oxygen. In the absence of oxygen also they can survive, and mycoplasma are pathogenic in animals and plants. So these are the main things we need to learn from kingdom Monera. So that's all about. Is it clear?
Yes, ma'am.
Okay, then let's move to next kingdom. Kingdom Protista. And compared to this, the basic characters are present in kingdom Monera. So compared to kingdom Monera, slightly evolved organisms are present in kingdom Protista. In this, most of them are unicellular. Most of the organisms are unicellular in kingdom Protista. But compared to that Monera, here we can see eukaryotic organisms also. From the chart itself, I mentioned, here you can see the eukaryotic organisms also. And the cells are developed. Compared to Monera, the cells are developed and most of them are aquatic. Aquatic forms are present. Most of them are aquatic. But terrestrial organisms are also present, but mostly they are aquatic. And everywhere it's present. Widely distributed, worldwide distribution is there. And Protista, they are, compared to Monera, they are slightly evolved and cells are developed. And then most of them are aquatic forms and eukaryotic cells are present. Then these are the organisms, they form phytoplanktons or zooplanktons. Then they are reproducing by asexual or sexual methods. And they have, I said aquatic, most of them are aquatic. So they have flagella or cilia for their locomotion. And they are eukaryotic. Eukaryotic. In the chart I mentioned, from there itself I explained it is eukaryotic. Eukaryotic means they have membrane bound nucleus and other organelles are also present. And photosynthetic forms are present. So chloroplast is present. And here locomotory organs, which are the locomotory organs, flagella and cilia. They are present in kingdom Protista. And next is, here you can see kingdom Protista. Kingdom Protista, it is the link between Monera and other kingdoms. It is coming in the middle of all these kingdoms. Like here, you can see in Kingdom Monera, some kind of disturbance from where it is. Okay, in Kingdom Monera, it is showing the basic characters, but here slightly old characters are there. So this is the connection, or it is showing the slow level evolution or growth to the next kingdom. Next is classification of kingdom Protista. All organisms which are coming under kingdom Protista, they are classified into different groups. First one is chrysophytes, then dinoflagellates, then euglenoids, slime molds, protozoa. Kingdom Protista is classified into chrysophytes, dinoflagellates, euglenoids, slime molds, and protozoa. In Protista, first one is chrysophytes. This group includes diatoms and golden algae. Diatoms and golden algae are included in chrysophytes. These are diatoms, and here you can see the golden algae. Here it is having golden pigment. This golden algae is having golden pigment which gives the golden color appearance. And they are found in freshwater and marine water. These are found in freshwater as well as marine water. These chrysophytes are present in freshwater and marine water. And size, they are showing diversity in size, and they are forming colonies, filaments, and the filaments are up to, some filaments are up to 2 meters. There in this side you can see all these things. Inside they are showing diversity, and they can form colonies and filaments. The filaments are up to 2 feet. And the types of diatoms based on shape, they're centric and pinnate. These are the two types. Note down this, two types of diatoms based on shape, centric and pinnate. And the golden algae, which is diatoms and golden algae, both are in this group. The present pigment, which is the appearance. And is, see, diatoms, the cell walls form two thin overlapping shells. Here you can see the overlapping shells are present. Two thin overlapping shells are present, like soap box. How a soap box it will fit together, same like that. Here two overlapping shells are present in diatoms. And the walls are embedded with silica. Note down, two thin overlapping shells are present in diatoms. Deposition in their habitat. And the accumulation of this will accumulate over billion of years, and it is called as diatomaceous earth. The accumulation for so many years it will get accumulated, and it is called as diatomaceous earth. This is not a single day collection, but billion of years it will get collected, and it is called as diatomaceous earth. And this soil seems to be gritty. That gritty soil. So this is used in polishing, filtration of oils and syrups. Here you can see in this have so many usages. So million of so many years it will get accumulated, and it is called as diatomaceous earth. It is the deposition of diatoms, cell wall deposition of diatoms. It is called diatomaceous earth, and it is used for polishing, filtration of oil and syrups. Then they have so many uses. See, here I have mentioned 40 plus for their. We are celebrating one day for diatomaceous earth day also getting celebrated, so we can make out the importance of this diatomaceous earth. In the previous one, you need to note down the diatomaceous earth. That is the main thing. And next is dinoflagellates. These appear in different colors and depend on main pigment present in them. Like yellow pigments are present, yellow, green, blue, red, all these colors are present. You can see here in diatomaceous earth. In dinoflagellates. And most of them are marine and they are photosynthetic. Marine and photosynthetic forms. On the outer surface cell wall, it has a stiff cellulose plate. You can see here it is a stiff type. You can see from these pictures it is stiff cellulose plate. Cellulose plate is present. Outer surface of cell wall has stiff cellulose plate. And these points you just note down. These are dinoflagellates. Yellow, green, blue, red, all these in each one different types of pigments are present, and stiff cellulose plate is present on the outer surface. Here you can see the exact structure. Dinoflagellates, you can make out from the name itself. Di means two. So two flagellas. As the name indicates, it is having two flagellas, and flagellas are not free. It's not free. And the flagellas are dissimilar. Here I have mentioned the flagellas are dissimilar. It is having two flagellas and which are not free, and the cell wall is stiff, and the cell wall is having silica deposition. Cell wall is stiff and it is having silica deposition. Dinoflagellates have two flagellas which are not similar, and the cell wall is having silica deposition, and the flagellas are not free. These flagellas, they are not free. You can see one flagella here like this and one is like this. One is present in the longitudinal groove and one in horizontal groove. One horizontal and another one is longitudinal. Here you can see one is here and one here. It is not free, it is in the groove. And red dinoflagellates. I said here so many pigments are present: red, blue, green, yellow. All these pigments are present in them. So red dinoflagellates, it is sometimes it will multiply rapidly and it will form the red color or red appearance to the seed. For example, for red dinoflagellates, gonolads. Note down the name gonolads. It is the example for red dinoflagellate. It will multiply rapidly, and the rapid multiplication may cause the red appearance of the seed. And some dinoflagellates produce light. Some produce light, and it is called as bioluminescence. They emit light. Some euglenoids. The third category in kingdom Protista, it is euglenoids. And in this, majority are freshwater forms. They are freshwater forms and mainly that is in stagnant water. It's present mainly in stagnant water. They are freshwater forms. and they don't have a cell wall. Instead of that, they have another layer which is rich in protein. This layer is called as pellicle. So majority are freshwater forms, and they don't have cell wall. Instead of that, they have protein rich layer which is called as pellicle, and this makes their body flexible. If cell wall is there, means it will be rigid body, right? If cell wall is not there, instead of cell wall, here they have a protein rich layer which is called as pellicle. That makes their body flexible, and they have two flagella, one short, long. One short and one long flagella they have. Next is, and they are photosynthetic. In presence of sunlight, they are photosynthetic. But when sunlight is available, they will perform photosynthetic. In the absence of sunlight, they act as heterotroph, or they eat some other smaller organism. In the presence of sunlight, they will act as autotrophs, and when it is absent, they will eat other small organism. They will act as heterotrophs. For photosynthetic, need a pigment which is present which is present in higher plants. So sometimes it will do photosynthesis, and sometimes they will act as a heterotrophs. Next category is slime molds. They are saprophytic parasites. Slime molds are saprophytic parasite.
Excuse me, ma'am. Ma'am.
Yeah, tell me.
Ma'am, can you hear me, ma'am? Ma'am, can we continue next class, ma'am? I have a school class now, ma'am.
Okay, it's already late, right? Okay. So you go through all these things, okay?
Okay, ma'am. Thank you, ma'am.
After this, it's clear, no?
Yes, ma'am. Yes, ma'am.
Yeah, okay. Thank you.